Like I told you in the last class that there are methylations and demethylations, acetylation and deacetylation. Accordingly, you had methyl transferases, demethylases, acetylases, histone acetyl transferase, and then deacetylases. Both kind of reactions. All these enzymes are actually in the system, they are together. In some of the lower organisms also this kind of H3K9 methylation and histone methyl transferases are required for DNA methylation. Here the Arabidopsis and Neurospora. There are other set of proteins called polycomb proteins, group of proteins, which target the gene repression marks to nucleosomes. You remember the repression marks H3, K27, Me. Me refers to that methyl group. If it is simply Me, it means only one methyl group is there. Me2 means it will be two methyl groups. Me3 will refer to three methyl groups. So, the methylation can be single or double, methylation, or it can be even triple. Three residues can be there. Three groups could be methylated on the same lysine. That is single, double or triple methylation on the residue. Have been implicated in targeting and maintenance of DNA methylation. Again, I mentioned earlier also that methyl cytosine binding proteins and the protein complexes can also bind to methylated CpG sites. Of course, ultimately it is a silencing signal. These complexes contain also HDACs. Just now I mentioned the acetylation, methylation, those enzymes involved in this methylation, demethylation, acetylation, deacetylation, they are all in one complex. That is what is mentioned here. Of course, some of these are highly characteristic features of the transcriptionally silent regions of the DNA. So, when these enzymes are there, they can silence the expression. Of course, they can also some of them can activate. So, the DNA N-methyl transferases also they interact with HDACs, which I have already mentioned. Changes in cancer include the increases in the levels and activities of DNA methylation catalyzing enzymes, or the proteins in the complexes that modulate the enzymes that catalyze transcriptional repression of the so called histone modifications. Loss of acetylation at H4K16, which means histone 4, lysine 16, and the increased levels of H4K20 acetylation are also thought to play a role in this. Then DNA methylation at gene promoters, this is an important thing. In the promoter, if there is hypermethylation, then that can lead to gene silencing. So, that is a signal, that is the most important epigenetic signal. So, histone modifications associated with this gene transcription such as acetylation of H3K9 and H4K16, they decrease. That is, here these are active in transcription, but then their levels decrease. So, that means transcription can be decreased. So, we are talking about gene silencing factors. One is DNA methylation, the other one is histone modification. Histone modifications associated with transcriptionally repressive chromatin such as H3K9Me2. Here you see the double methylation, 2 methyl groups, Me3, 3 methyl groups. So, and H3K27Me3, they increase. So, all these are repressive marks. So to silence a gene, you can have all these marks. That is, you can have DNA methylation at the promoter level, hypermethylation, and you may also have histone modifications, acetylation at the residues shown here, and so on. Not only simply the acetylation which indicates active transcription, those residues will be less. That is one way. The other one is the repressive markers may increase. So it is a dual action. Repressive markers increase, the active markers decrease. Transcription, those changes which result in increase in transcription, they decrease, and those which repress transcription, they increase. So that is how this histone codes work. So this way you can ultimately suppress a gene. So that is the idea, because this is very important, because I told you this is the major mechanism by which tumor suppressor genes lose their expression in cancer. So that is an important mechanism. H3K27Me3 histone modification is an important indication in human embryonic stem cells for genes that would become hypermethylated in adult cancers. Additionally, these are some examples. H3K9Me2 and H3K27Me3 may also be involved. Just remember some of these residues, because these are some important markers. In fact, people have generated maps based on these methylation marks and histone modifications, acetylation and all that. There is a big map. They develop atlas and so on. It's difficult to remember all of them, just remember a few of them should be fine. At least have some examples for activation of transcription, some examples for repression of the transcription. That should be fine. So during cancer development, at any stage these changes may happen. You may ask, sir, when this happens? It can happen usually at the initial stages, but practically it can happen at any stage. One important difference that you must note is, while mutation is almost a permanent change, but these epigenetic changes are reversible. You can control the expression for a particular environment, because they are reversible. After all, methylation, if it is methylated, if you have the other enzyme active, you can demethylate it. If it is acetylated, you can deacetylate it. Then you can switch on or switch off. So that is very important point you may have to remember. Such a change may confer the ability to withstand adverse circumstances such as chronic inflammation, reactive oxygen species, or even the other condition, hypoxia, etc., and still make a progress towards carcinogenesis. So you can regulate various factors that may ultimately favor the progression of the carcinogenesis. By playing with the epigenetic changes, you can ultimately make the cancer cells progress towards their development. So that is the idea. Of course, you can do the reverse also. Cells encountering DNA damage will normally undergo apoptosis, but if these changes in methylation occur, they may survive, for example. So this is why we are writing, we are talking about these things, is because this can serve as a mechanism for tumor progression. You can silence certain genes and that might help in tumor progression. For example, if you silence the tumor suppressor genes, that will help in cancer progression, simple. So that is what is shown here. One more example, P16 gene is a frequent target for early methylation in breast cancer and non-small cell lung cancer. The previous slide I was just telling the general mechanisms. I am giving you some examples now. Histologically normal mammary epithelium without malignancy can harbor P16 promoter hypermethylation. Experimentally, early loss of P16 in mammary epithelial cells precedes genomic and epigenetic instability. Genomic instability is a characteristic feature of cancer cells. So, this can be one example. Another example is in colon cancer, aberrant crypt foci show DNA hypermethylation of what are known as SFRPs, which are, we have heard about them, secreted Frizzle receptor proteins. Where did we hear about them? In Wnt signaling, because they are the receptors for Wnt ligands. So, Wnt interaction with its receptors, so that can be altered. If this hypermethylation persists throughout colon tumor progression, if you may remember that I told you Wnt signaling is very important for the pathology and tumor progression in colon cancer. Wnt signaling is very important in colon cancer. So, this can help in tumor progression. It may drive the Wnt pathway further in collision with the other mutations. It is not only one change, there must be several changes before a normal cell gets transformed into a cancer cell. So, these are important changes. Since we are talking about methylation and other epigenetic changes, we are giving these examples. So, understanding these kind of epigenetic changes obviously may provide potential biomarkers for risk assessment, early diagnosis, and also for prognosis assessment. So, this is what usually people do. What do you want? You want to know whether a cancer will progress. You want to see whether there is progress in the treatment. After treatment, what is happening? So, you want to look at that, or ultimately you want to see what are the risk factors involved. So, for all these processes, epigenetic changes might help. They may play a role, that is what it says. H3, histone 3 and histone 4 modifications correlate with aggressiveness in the case of prostate cancer. So, that is one example. Several examples are there. See, one more example, they have given DNA methyltransferases and histone methyltransferases for H3K27 methylation and other polycomb group gene silencing constituents. I already talked about polycomb proteins. So, these are some examples. Reversal of DNA hypermethylation and aberrant gene silencing are also helpful, because if you reverse, I told you it is reversible. So, can you reverse that by some strategy? That can also help, because you are then making the tumor suppressor genes to be expressed. These changes have suppressed their expression. Now, you reverse these changes, thereby you allow the tumor suppressors to be expressed. So, that can be one strategy. After all, we are studying all these things ultimately to develop strategies to treat cancer. So, you have to think on those lines whenever you study cancer biology. You should think, if you understand certain things in cancer, then the next thing you are going to think about is how to make use of this to treat cancer. So, that is how, that will be the line of thinking in this whole course. One idea is to promote, to use the promoter hypermethylation sequences as a biomarker for each type of cancer. What it means is, in each type of cancer there may be certain hypermethylations. I think I have given some examples also, some table in the last class. I mentioned that there are certain genes which are, whose promoters are hypermethylated, BRCA1, all those things. I have given some examples. So, that can serve as a biomarker. If that is hypermethylated, there are methodologies to understand whether a particular gene promoter is hypermethylated or hypomethylated. There are various methods, bisulfite method, sodium metabisulfite method, and all that. There are methodologies. So, you will be able to see that. See here another example, sputum DNA from patients at high risk for lung cancer have shown that invasive tumors may be predicted more than a year before clinical detection of the cancer. This is a very important contribution, because more than 1 year before they develop aggressive cancer, you may be able to detect it if you just take sputum and then test for these markers. Abnormal methylation markers in sputum were also useful in surgically resected early stage lung cancers. What is the gene? P16. P16 gene in DNA from lung cancers predicts poor outcome. So, that is one example. Then in normal colon tissues, gene promoter methylation increases with the age and parallels the risk of cancer. This is another just an observation. Metastatic potential can be predicted on the basis of E-cadherin promoter methylation in breast and oral cancers. This is another example. One more example, you remember I told you guanosine also can be methylated, right? O6 methylated guanosine methyl transferase is an enzyme, is a gene that encodes for a protein that mediates removal of bulky alkylation adducts from guanosines. So, this is one simple example. I think I will talk about this later also in DNA repair mechanisms, we will talk about that. Silencing of this particular enzyme is observed in many cancers, especially I think in brain cancer. If this happens, consequently these cancer cells cannot efficiently repair alkylation damage. When we talk about DNA damage, DNA repair, we will talk about this. So, such cancer cells are sensitive to alkylating agents. Actually, if you have this defect, then what happens? This methylation, if that is happening, then what happens? The cells are actually sensitive to drug. Although it is a aberration, it is an aberration, but that can help in sensitizing the cells to certain anti-cancer agents, alkylating agents. One example is that temozolomide, which is usually given in brain tumors. Brain tumors harboring O6 MGMT respond better to temozolomide. So, this is one example. So, ultimately the idea is to develop the knowledge we gained for either prevention or for therapy. I already mentioned that mutation is not reversible, but epigenetic changes are reversible. You can also combine individual or combined targets for interventions. You do not have to go through only one change. You can combine 2-3 changes. That also I have mentioned how that methylation, acetylation and other things. If you know the theory behind it, you might look for suitable combinations for a particular strategy. So, that is what they have just mentioned this. We will proceed with other agents. For example, DNA methylation inhibitors, 2 examples are given here, 5-azacitidine and 5-aza-2'-deoxycytidine. Both are basically demethylating agents. The efficacy of these so called azacitidines that correlates with acute reversal of gene silencing. DNA methylation inhibitors are usually some nucleoside analogues. So, they can be converted to deoxynucleotide triphosphates and can be incorporated into replicating DNA in place of cytosine. We will, I think, did we complete that? I have said HDAC inhibitors, did I say anything about that in the previous classes? Molecularly targeted drugs, I think we have discussed that. That is why I have written here. I have mentioned about these drugs, HDAC inhibitors and all that. Probably I must have mentioned. But you see the idea, ultimately it is halting the DNA replication. So, that is one way of treatment. That is what they can be converted to deoxynucleotide triphosphates. So ultimately, if they are incorporated into the DNA, then DNA will not be able to replicate because it's a different base. HDAC inhibitors are already in the clinic. Of course, the most important one I will discuss that. It is called SAHA, suberyl anilide hydroxamic acid. Commercial name is Orinostat. This is the most important thing. It has been approved for the treatment of T cell cutaneous lymphoma. What they generally do is that the HDAC inhibitors, they can have anti-tumor, growth inhibitory or pro-apoptotic and pro-differentiation properties. These are incidentally, these are the most important properties if you want to develop a treatment strategy, because you can treat cancer by inducing differentiation, you want to inhibit growth, you want to induce apoptosis, right. These are the important ways by which you will be treating the cancer. Of course, these inhibitors, they upregulate the expression of P21. If you remember, P21 is a cell cycle inhibitor and the mechanism is hyperacetylation of their promoter by these drugs. So, these drugs basically because HDAC inhibitor, what is meant by HDAC inhibitor? Histone deacetylase inhibitor, right. So, if deacetylation is inhibitor, what is happening? Acetylation goes up. If you are inhibiting, it is a balance between acetylation and deacetylation. If you are inhibiting deacetylation, you are enhancing acetylation, simple. So, that is what is happening, acetylation of its promoter by these drugs. That is one example. Another example is trichostatin A. I want you to remember this. This is a derivative of this SAHA, trichostatin A. The commercial names you do not have to worry, panobinostat and belinostat and all that. Apart from these, some small chemicals like sodium butyrate, it is a short chain fatty acid, phenylacetate, phenyl butyrate, valproate, they can also act as HDAC inhibitors. But the major HDAC inhibitor or the first HDAC inhibitor discovered was SAHA, suberyl anilide hydroxamic acid. So, that is very important to remember. There are also other compounds like some cyclic peptides, benzamide derivatives, which can also act as HDAC inhibitors. Here we are just talking about HDAC inhibitors because they are already approved in the clinic. So, HDAC inhibitors are important class of compounds. There are other compounds because we have talked about methylation, demethylation, acetylation and deacetylation. So, any compounds which we interfere with these processes will be useful in one way or other. So, that is what we are showing here. Inhibitors of demethylases. Some examples are given here: pargylene, phenelzyne, tranyl cypromine. So, these are some examples. The inhibitors of methyl transferases, chatosine, DZNEP and alantodapsone. Names are a little bit tongue twisting, but just try to remember some of them at least. The last one is very interesting, inhibitors of histone acetyltransferases, HAT as we call them, histone acetyltransferases. They include some of the well known compounds like curcumin, garcinol and anacardic acid. These are from natural sources. So, one more important compound is called sirtuin. A lot of people are doing research on this. HDAC inhibitors, there are so many classes, class 1 HDAC inhibitor, class 2, class 3 and so on. This belongs to class 3 HDAC inhibitor. One example that sirtuin is class 3 HDAC. Sirtuin means it is a HDAC, class 3. Its inhibitor is called sirtinol. This is gaining importance since they can induce p53 activity, and ultimately that can induce apoptosis. So, it is basically in the context of inducing apoptosis, these compounds are very important. The sirtuin, there are many commercial companies are interested in synthesizing these kind of compounds and marketing them also, because they are found to be very effective inducers of apoptosis ultimately.